Okay, I think it'd be good to get started. I just want to welcome everybody to the spring seminar series for the Healthy Aging at Tufts University Priority Area Research Group. This is the last seminar for our spring series. And I just want to remind everybody that we do have an upcoming mini retreat, which will be held in May, and we'll be sending out an announcement for the mini retreat, which will be focused mostly on internal aging projects at Tufts. So it's just my pleasure now to turn over the moderator duties to Dr. Alan Taylor to introduce today's seminar speaker. Thank you, Alan.
Thank you so much, Roger. And it's not just a duty, it's an honor to introduce Dr. Toren Finkel today. I've known Toren Finkel's work for several decades already. He's really a legend in his own time. Dr. Finkel received his undergraduate degree in physics and his MD and PhD from Harvard Medical School. Following a residency in internal medicine at the MGH, he completed a fellowship in cardiology at Hopkins. In 1992, after completing his clinical training, he went to the NIH as an investigator with the intramural research program of the NHLBI, the National Heart, Lung, and Blood Institute. Over the next 25 years at NIH, he held various positions, including the chief of cardiology branch and the chief of the Center for Molecular Medicine within NHLBI. He's a member of the American Society for Clinical Investigation, and he's also a member of the elected member of a very prestigious National Academy of Medicine. He serves on numerous editorial boards, including currently serving on the board of reviewing editors for science. Dr. Finkel assumed the role of the director of the Aging Institute and the Nicholas Beckwith and Dorothy Beckwith endowed, Nicholas Beckwith III, excuse me, and Dorothy Beckwith endowed chair of translational medicine at the University of Pittsburgh. His laboratory has made fundamental contributions in our understanding of the role of reactive oxygen species in mitochondrial function in aging and age-related diseases. And it's through those fundamental discoveries that I met Dr. Finkel many years ago. According to Google Scholar, he currently is one of the highest cited authors in both cardiovascular disease and in aging. He's also recently co-developed several small molecules which are anticipated to be in phase 1 testing within 12 months and may be of potential benefit for a range of age-related disorders. And I think together, this constellation of talents and contributions makes him extremely attractive and intriguing person to listen to his talk. And now I'll turn it over to you, Dr. Finkel. Thank you so much for honoring us with your time and presence.
Thanks so much, Alan. Thanks so much, Roger. It's really a pleasure to, well, not to be there, but to speak with all of you. Hopefully over the next 40 or 45 minutes, I can give you a little sense of, what both my lab is interested in and what the larger Aging Institute that I work at and direct at the University of Pittsburgh is sort of focusing on to sort of translate the biology of aging into new therapies for patients. And so just as a way of disclosure, since I am going to be talking about some of the small molecules that we developed, I just want to disclose that I co-founded with my colleagues at Pitt Generic Pharmaceuticals, and I serve as the CSO for that company at this time. So over the last 3 years at Pitt, we've been able to develop an Aging Institute, as shown here, with now about 13 or so principal investigators who really are trying to work on sort of this geroscience concept of trying to understand the basic biology of aging, with the idea that those insights will result in better understanding of aging, but also translation to sort of treatments for age-related diseases. And the goal of the Institute really is to understand the basic biology, but we've tried to be very translational focused. And so we have, and as you see, we have within the Institute the capacity to do high throughput drug screening. And so we have a robust capacity to do that. And that's resulted in some small molecules that I'll talk a little bit about, that we're very excited about, but and a variety of other projects that allows us, to reduce some of these concepts to drugs and the hope to do that. And we also have a desire to translate this, not only our own drugs, but other potentially repurposed drugs to sort of tackle aging biology. And I'll go through some of the work that our clinical focus is on in terms of testing neuroscience principles in humans. So I always like to start off, why we should study aging. And again, I think since this audience is particularly interested in therapeutics and translational potential, I'm going to use an example that I like to use, which, based upon the fact that I was trained as a cardiologist, just to give people a sense of the magnitude of aging's effects in human disease. So as many of you know, there's been a lot of epidemiological evidence that have defined various risk factors for disease, and particularly for cardiovascular disease. Studies like the Framingham near you guys have really been instrumental in sort of outlining risk factors for this condition. And those can be sort of quantified. And so in fact, if you have elevated cholesterol above, a total cholesterol above, for instance, 280, then your relative risk for a cardiovascular event doubles over the course of a 10-year period. Similarly, if your blood pressure is high and elevated, you roughly triple your risk. Diabetes doubles your risk. Smoking doubles your risk. But if you're a man age 70 or above, your risk of cardiovascular disease now goes up 7-fold. And so those are the sort of relative risk of age versus these other, what we could call conventional or modifiable risk factors. And so there's been, as you know, an incredible amount of effort and work has been done to sort of attack these risk factors of cardiovascular disease by lowering cholesterol, for instance, with statins, lowering blood pressure with a variety of different medications, treating diabetes, hoping to get people to stop smoking through a variety of means. And the composite of those interventions on these risk factors has been to really dramatically reduce the incidence and mortality from cardiovascular disease. But obviously, if you look at this chart, what's left on the table as sort of the elephant in the room is this idea of age itself. And given the sort of success in reducing these what's the modifiable risk factors, if you believe the concept that age is modifiable, then the impact on a disease like cardiovascular disease would be tremendous if you could, in a sense, modify age as yet another risk factor. And that's sort of the working hypothesis that I think a lot of people in the field are doing, that the tenets of aging biology will lead us to ways to modify this biggest of all risk factors for age-related diseases. And in fact, it's, as you can imagine, this is just not for cardiovascular disease. So every disease that we worry about, including cardiovascular disease, what's shown here in the top left, it's Alzheimer's disease, and below is cancer, that all of these diseases really have the same sort of age-dependent incidence, so that their incidence is quite low when we're young, and then sort of exponentially rises as we get older. And the concept here is that I like to think about it, some sort of very sort of Cartesian sort of XY coordinates, is that what most people are focused on is the y-axis of these diseases, thinking about therapies that will specifically affect cardiovascular disease or neurodegenerative diseases or cancer. So focusing on the y-axis. And I think what people in aging biology are doing is saying we want to focus now on the x-axis. That is, try to think about therapies and medicines that slow the progression of age as a means of shifting these curves to the right. And the idea would be that if you were successful, you could shift all of these curves to the right, because the x-axis is sort of common to all of these conditions. So the question is, how far can we shift these curves to the right? How long can we live? How long can we slow down? How best can we slow down the aging process? And I like to show this picture of probably the poster child of aging biology, which was Madame Jeanne Calment, who lived in southern France. And this is a picture of her when she was 121 years old. She lived almost 2 more years. And she had a really lovely life. I mean, nothing remarkable in terms of achievements except for longevity. But she lived in Arles, which is a reward unto itself. And her uncle had this paint supply store. So she got to meet when she was growing up, all the French Impressionist painters of the era, because they would stop in the store, and she had very complimentary things to say about Cézanne and not so many complimentary things to say about Van Gogh, but she lived a very interesting life. And the question is, did she do anything that we could emulate to sort of live as well this type of long life? And the answer is no. This is her as well. Smoking, which she did up till age 119, and she drank every day, and she did all the things that we're not really supposed to do. And in fact, you know, if you look at her children, they did not live exceptionally long. And in fact, genetics of longevity are quite controversial, and people give you different numbers. Some will argue that, you know, maybe our inheritance of lifespan is about 20 or 25% genetics. Other people will say, for most of it, it's even lower than that. And so there are other things besides genes that sort of determine our lifespan. And one of them that's been very interesting to us and others is the environmental factor of nutrient availability. And so it's been known for roughly 100 years that if you reduce the supply of food to animals like rats or mice, that they live longer. And that phenomena of caloric restriction or intermittent fasting, or all the sort of derivatives of this concept, is really a pretty evolutionarily conserved phenomena, and you can see it in some form or another in organisms as simple as yeast, and clearly up to mammals like mice and rats, and evidence suggests that it might be conserved in primates and humans as well. And certainly in rodents, it's a pretty robust phenomenon that you can extend lifespan 30 or 40% in at least some species of mice by reducing food intake. And so that phenomena really, as well as the sort of revolution in genetics, I think really allowed aging to be beginning to be parsed apart. And it really came not, I think, from the study of mammals, but really from simple organisms. People like Tom Johnson and Cynthia Kenyon and Gary Rufkin and others have used C. elegans really to really dissect the sort of pathways that regulate lifespan, and really develop simple models in which a single genetic mutation in an organism could change the median and maximum lifespan, and to realize that those pathways that were regulating lifespan in those organisms were highly conserved across evolution. And that in turn has allowed people to begin to dissect, you know, how these phenomena like caloric restriction might be translated molecularly into a change in overall lifespan. And summarizing really lots of people's work, I think a number of evidence point to that caloric restriction in organisms where it extends longevity is working through at least two separate pathways. One potentially is through these family of NAD-dependent deacetylases, the sirtuins, which my lab has had some interest in, which are regulated by NAD and hence potentially by nutrients, and which appear to impart some changes in overall lifespan. And the second pathway is this energy sensing kinase in the cell called Mtor, which is again regulated by nutrient availability, and which again appears to mediate overall lifespan. And so, as I said, we've been interested in both of these pathways, but I just wanted to show as a sort of a concept, a proof of principle work that we did a while ago on this Mtor pathway, just again to show you, I think, in my mind, the sort of this idea that aging is in fact regulated. And then because that is such an important concept, I think to sort of stress. I mean, I think when I was sort of in graduate school and medical school, I just viewed aging as sort of a random and stochastic degenerative process with sort of without the sort of biological regulation that you would think of most processes. And I think the work that really started in worms and has moved to mammals and hopefully will eventually move to human really suggests that that's not the case, and that like every other biological processes, aging is regulated. And I think the implications for that, I think I'll try to show you, is that we can then begin to think about intervening in these pathways and developing small molecules or other approaches that might regulate these central pathways linked to aging as a means of treating therapies. And so just as a way of an example to show you that regulation, I want to show you, go through very quickly a mouse model that we worked on a while ago that result in a reduction in overall mTOR expression. And this was really work done initially by a colleague of mine at the NIH, Beverly Mock, who generated these animals. And for reasons I won't go into, Beverly's lab had made a mouse, which we call the delta mouse, in which by gene targeting, neomycin cassette had been inserted into an intron of the mTOR locus. And this resulted in no real coding changes in mTOR, but it affected the transcription and translation such that that mice that expressed two of these that resulted sort of in hypomorphs of mTOR expression. And so if you obtain two of these delta alleles, you express roughly 25% the overall level of mTOR. Although it was normal mTOR, it was reduced to about 25% of wild type levels. And these mice are born relatively normally. And as you can see on the right, they look relatively normally. They're a little smaller, which is consistent with sort of mTOR's role in overall cell size. But besides the overall size difference, you really couldn't tell any sort of other distinguishing characteristics of these mice that express less mTOR. But if you looked at them long enough, and this was work done by a very talented postdoc, Julie Wu, in the lab, and followed them over the course of several years, what you observed was that they lived longer, and they lived substantially longer. These are males and females cohorts, and these mice lived about 20% longer than that had sort of reduced mTOR than their wild type controls. And we did a bunch of, phenotyping in these animals, and I won't go through all of it, but shown below on the bottom is sort of an example of memory and cognition in these animals. And I think and what it shows is that as animals get older, their ability to return by this test to remember, to learn new things is impaired. But the mice that had a reduction in mTOR, that age-dependent impairment, which was less evident as they got older. And we did a bunch of sort of in vivo phenotyping of age-related parameters. And not all, it turns out, but many of the sort of age-related declines that we saw in the wild type looked like they were significantly slowed in these hypermorphic mTOR mice. So not only did these animals live longer, is what I'm trying to say, that they had a longer health span. So in tissues and organs where we saw age-dependent decline, for the most part, as I said, that appeared to be slower in these hypermorphic mice. And so the end result is that by tweaking one out of 30,000 genes, in this case mTOR, by tweaking it and not even getting rid of it, but just by reducing it by to a quarter of its normal expression, we were able to change the sort of lifespan of the organism by a significant amount. Again, so 20% increase in lifespan, if extrapolated to humans, would be changing our median lifespan from 75 years to 90 years. Again, by changing one out of sort of 40,000 of our genes. And not only, if this again could be extrapolated, not only would you expect to live longer, but you would be able to think better and do all the other things that you would like to do throughout your lifespan. Again, suggesting that, in fact, that going along and affecting that x-axis that I brought up before might be potentially doable. And so that's been sort of, I think, what has sort of galvanized us to try to think about ways of intervening on the central biology of aging to develop new medicines that might be useful across a wide range of age-related diseases. And so I'm going to tell you one aspect that my colleagues and I have done at Pitt, and that, as I said, we are in the process of bringing to human trials, hopefully this year. And that is another pathway that we've been very interested in. We think is really important in overall lifespan, which is the process of autophagy that many of you know about. And the reason we were sort of interested in this pathway as a potential druggable way is if you look again at the myriad of interventions that have been shown in organisms such as worms to regulate overall lifespans. And so, as I said, dietary restriction is one, but in the worm, even things like germline removal or impaired mitochondrial function or reducing mTOR signaling or TOR signaling or reducing insulin signaling have all resulted in worms that live longer. And these are very disparate sort of interventions. And so people have searched for what could be a common downstream mediator. And I think, not, with 100% certainty, but in most of these examples, as shown here, what people have seen is that all of these life-extending interventions produce or maintain an increase in autophagic flux as the animal get older. And in many of these cases, in genetic sort of epistatic experiments, they've been able to show that if they impair autophagy, that one can in turn one doesn't see the life-extending effects of these interventions, suggesting that autophagy is both a necessary and potentially sufficient requirement for many of these life-extending interventions in lower organisms. And so it would suggest that if you could increase autophagy, that might be in fact very beneficial for a wide range of diseases. And so if you think about how that could be done, there certainly are a number of ways, but one of the things that we sort of honed in on, my colleagues and I, was this transcription factor called T-Fab. And T-Fab, as you know, is normally tethered to the lysosome under normal conditions. And then a variety of lysosomal stresses or nutrient stresses causes T-Fab to go to the nucleus where it binds to DNA and then acts as a transcription factor to turn on roughly 400 or so genes. And so and those genes are involved in the making of new autophagosomes and the making of new lysosomes and really everything that's required in terms of transcription and proteins in this sort of whole autophagy lysosomal pathway. And so, by turning T-Fab on, one really augments the capacity of the cell to increase its autophagic capacity and to potentially improve, we thought, a variety of age-related phenotypes. And so there's been a lot of evidence to suggest that, again, augmenting T-Fab may in fact be very beneficial. And here's just a lot of papers that have sort of shown that. And in general, there hasn't been a small molecule that could do this with high efficiency. And so most of these studies as shown here, with one exception, are based upon sort of adenoviral delivery of T-Fab to sort of, for instance, the CNS as a way of increasing autophagic flux for diseases such as Alzheimer's or Parkinson's, in which protein aggregates accumulate and which an increase of autophagic flux might be beneficial. And so what my colleagues and I have done, and my colleagues Bill Chen and Yuan Liu, was that we were able to develop a small molecule that activates T-Fab. And here's some of the evidence of the use of that small molecule. So to the left is a panel of gene expression of known T-Fab targets in cells treated with one of our earlier compounds. And to the right is evidence of lysosomal biogenesis in cells, which is sort of an increase in overall lysosomal number in the setting of increasing concentrations of our clinical candidate, in this case, what we're calling GT300. And so we see a nice increase in the actual numbers of lysosomes in the cell when the cells are exposed to this compound, which is really a really remarkable, in my mind, still a remarkable phenomenon, because again, the idea that a small molecule could produce a new organelle in the cell is really exciting, I think, in terms of the ability to think about how this might be useful, therapeutically. So, obviously one of the things where increasing autophagy could be beneficial, as I said, or in diseases in the central nervous system, in which mutant proteins aggregate and accumulate and appear to have toxic effect. And there clearly is lots of human genetic evidence suggesting that impairment in the autophagy pathway might predispose to these aggregate proteins, and that if you can increase autophagic flux, and as I showed you some examples where people have overexpressed T-Fab, but any sort of way that you can increase autophagy is thought to be beneficial as a way of clearing these aggregates and potentially helping these patients. And so we've been engaged over the last year or two with colleagues at the University of Pittsburgh. And so this is work to the left of Emily Roca and Tim Greenemeyer at Pitt, who've taken some of our early compounds and looked to see their effects in animals. And what Tim and Emily have seen is that, like I showed you in cells, that these compounds are able to induce lysosomal biogenesis in animals. And it looks as if that induction of autophagy is able to reduce, in this case, the burden of endogenous synuclein in animals that are elderly. And we've gone on and shown, as shown on the right, in a model where we inject, in this case, synuclein fibrils, that these small molecules might be able to help, in this case, improve the motor phenotype, the Parkinsonian motor phenotype that one sees in these animals, all suggesting that this concept might be tractable in that by activating T-Fab, we can increase the number of autophagic flux and increase the number of lysosomes. And that in turn might be beneficial in sort of the clearing of aggregates in the brain or other things. And so, as I said, we're very interested in this concept. This small molecule is actually scheduled to begin phase 1 testing in humans at the end of the summer. And we're still, we're sort of thinking that it might have lots of implications and applications in human diseases. I sort of sketched through our thoughts on sort of central nervous system, but it turns out that there's lots of other areas where autophagy and quality control are really, really important. And obviously other areas in which lysosomal function is really important. And so we don't actually know where this concept will have the most therapeutic application. And so we're still sort of pursuing many of these models in animal models to sort of try to hone in on where their clinical end result will be the most beneficial. But I do think it's, I think we're very excited by this idea that, you know, we can begin, at least there's a potential strategy to sort of take sort of these central mechanisms of aging, like autophagy, and reduce them to sort of small molecules that might be of benefit. And we'll see if this molecule is beneficial or not and safe or not. But and so I'll hopefully come back in a year or two to share our progress. But obviously, autophagy, you know, it represents sort of one aspect of the sort of hallmarks of aging and sort of the proteostasis hallmark. But there's a lot of other hallmarks of aging where this concept, where one can begin to think about intervening using small molecule approaches as a way of helping patients with age-related diseases. And so I'd like to go through another strategy that my lab has taken recently in the area of senolytics and how we're approaching developing molecules that might be useful as senolytics. That might be a little different than others have done this. And so I know you've probably heard a lot of this already because I saw who's spoken already, and I don't want to repeat too much of what you probably all are now experts on, but there is this idea that cells, when are damaged, really have several fate choices. I like to think about it. So with obviously, if the damage is not so severe, they can repair. But if the damage is significant, you know, the damage can be unrepaired, and the cells can undergo apoptosis, or the cell can adopt another cell fate, which is to go into permanent cell arrest and undergo senescence. And actually, the reason why the cell chooses one or the other is not entirely clear to me. But I think for many people, the idea that senescence represented a sort of tumor preventive mechanism, so that cells were damaged but couldn't divide, and therefore these cells, you know, this was a good thing for you. And I think there is still a lot of evidence to suggest that's true, but I think obviously the work of Judy Campisi and others have suggested that these cells, although not dividing, clearly are not inert, and they can secrete a lot of things, as Judy's called the senescence-associated secretory phenotype. And these factors that emanate from the senescent cells can really change the microenvironment and have also distal effects that can predispose to age-related diseases. And a variety of evidence that suggests that sort of in Koch's postulate that, if you remove these cells, senescent cells through a variety usually of genetic means, that, that makes certain types of animals live longer or healthier. And similarly, if you take healthy animals and give them senescent cells, you can sort of mimic age-related diseases, all suggesting a sort of causal relationship between senescent cell burden and age-related diseases. And then there's the work that you've heard from Jim and others, Kirkland, that you can potentially pretend to think about developing small molecules or combination of small molecules that could specifically target senescent cells and remove them, hopefully in a sort of therapeutic fashion, without affecting the normal cells, much like chemotherapeutics target cancer cells and don't target normal cells, or at least have a wide enough therapeutic index to be useful on that. And those senolytics would be useful. And again, there's a lot of support that senolytics, either again genetically encoded through sort of tricks or through small molecules, really are very useful in a variety of preclinical models in terms of reducing age-related phenotypes of a wide range of diseases. So, and this is, as you probably heard from Jim, has already gone into clinical trials, and so there's been, this is I think from Jim Kirkland's work, trials in diseases like idiopathic pulmonary fibrosis, which is in the inherited form, is a disorder of telomere maintenance, and there's an accumulation of senescent cells in the lung, and so people are beginning to sort of use agents as senolytics to see if they're safe, and with the hope that if they are, that they can potentially be these trials can be expanded to look for sort of efficacy symptom signals. I think, at least from my looking at the landscape, what most people have done for senolytics to date is to repurpose agents, mostly from oncology. So agents like navitoclax, which were an oncological agents that target BCL2 family members, or other agents like this have been used and sort of tested to see whether or not they have a sort of preference or a therapeutic index in killing senescent cells while not killing normal cells. And so that's certainly a very fast way of getting into the clinic, because these drugs have already undergone a lot of safety testing and FDA approval. But I think one might argue that this universe of drugs that are FDA approved that you want to repurpose may not represent the ideal way of attacking senescent cells. And so the question is, can you use basic biology to try to understand how you might develop senolytics from a sort of more sort of unbiased approach, and not use repurpose agents, but really identify sort of the unique vulnerabilities of senescent cells through genetic means. And so this is the work that's been done in the lab by a talented MD PhD student in my lab, Shahini Ghosh Chaudhry. And it really is based on this concept of synthetic lethality, which is really a concept that has emerged in many contexts, but therapeutically in cancer biology. And so basically, it's sort of outlined here. If you have a phenotype or a process, that both protein A or pathway A and pathway B contribute to, that process is pretty okay if you get rid of A. It's pretty okay if you get rid of B, but the combination of A and B is lethal, basically. And so this has been sort of used in cancer biology. So for instance, if you have a BRCA1 mutation cell or tumor that's BRCA1 deficient, and BRCA1 is very important in DNA repair, and particularly in homologous recombination of DNA, then that represents pathway A, and then if you hit them with, for instance, a PARP inhibitor, which is also involved in DNA repair processes, that although the PARP inhibitor alone is not so lethal, and BRCA1 lesion alone is not so lethal. The combination is lethal for those cells. And so that has led to the use of PARP1 inhibitors clinically in patients with BRCA1 deficiencies. And so it's uniquely synthetically lethal for that combination. And so the question is, can you find genes that are synthetically lethal for the senescent state? And so the idea would be that what you're looking for are genes that, when deleted, have really no effect on normal cells, but when they're deleted in the context of senescence, are lethal. And so that the identification of those genes then provides a target list, because then a small molecule that inhibited those genes that are synthetically lethal in the senescent state would reproduce this screen, which I'm going to go through, which is a CRISPR-based, whole genome CRISPR-based screen to look for synthetic lethality. So that's sort of complicated, but maybe I can walk you through how it works. And again, this is ongoing work, so we don't have the sort of definitive answer, but I just wanted to go through the approach because I think it's just, in my mind, an alternative way of trying to develop senolytics, which we're sort of interested in doing. And so this is a cell line that's a little complicated, but it's an immortalized mouse embryonic fiber line that we've developed that has, in this case, a flocked allele for the telomere binding protein TRF2. It also has an estrogen responsive Cre recombinase in the cell line, as well as Cas9. Okay, so these cells are normal and grow and quite well and are perfectly fine. But if you add tamoxifen to these cells, the Cre then translates to the nucleus. It deletes TRF2, which is a telomere binding protein, and it produces a telomere uncapping in these cells, and they undergo rapid and sort of uniform senescence. Even though their telomeres are long, they're uncapped by the deletion of TRF2, and that causes these cells, which are immortalized, to undergo a sort of growth arrest over a sort of 5-day period and exhibit all the attributes of senescence. Okay, so the way we did this is then we took these cells and infected them with a whole genome CRISPR library of mouse genes, so that we could, basically knock out, using a sort of MOI of like 0.3, that we would have sort of a knockout library of cells in this population. And so this library has 60,000 guide RNAs and covers the entire genome of the mouse. Okay, so now these cells undergo puromycin selection, and so the guides are then selected for. And so we have this population of cells now that has CRISPR, whole genome CRISPR knockout. And then what we do is we split the population, and in half the cells we give tamoxifen and induce senescence, and half the cells we let them continue to grow in the absence of tamoxifen. And in this case, we waited 6 days or 12 days, let's say, and we collected DNA in both populations. And so, and then we turned we sequenced the DNA, and then we turned it over to a bioinformatics person, and we said, find us guides that are missing in the senescent cell population that are abundant in the control population. So these are genes that, when knocked out, so the guide being present means the gene is knocked out, so that the normal cells are perfectly able to tolerate knockout of this gene, but the guides are missing in the senescent cells, meaning that the senescent cells presumably were not able to tolerate deletion of the gene in the context of senescence. So that those guides represented genes that could be synthetically lethal. So I won't go through, we have a list of about 20 genes or so that fit that product, and we're going through them. I just want to show an example of one. This is E3 ubiquitin ligase that we've done a little bit of work on, and just to show you the sort of phenotype. So on the top row is genes where we've gone ahead and knocked out this E3. And the bottom row is sort of an untargeted CRISPR. And then in the first row is without tamoxifen. There's no effect of knocking out this gene in the presence, and when the cells are not senescent. And then when we make the cells senescent, they undergo a morphological change you see on the bottom, but in the top, you can see the cells are starting to die. So again, we're knocking out this gene in the normal context has no effect, but knocking it out in the setting of the senescent cell produces cell death. And so that suggests that if you had a small molecule that, if we can confirm it with other studies, that if you have a small molecule that could inhibit the effect of this gene, then it would produce synthetic lethality potentially in vivo, and it would be a potential senolytic therapy. And so this CRISPR screen thereby provides us really a target list for senescence or for senolytics. And so we're very excited about, this is probably not a good one because that's not a particularly easy gene to target, but we have at least some candidate kinases that could fit the target of potential, druggable molecules. And so we're very eager to sort of pursue that. The other thing that this cell line and this types of screen can do, and again, we're very interested in drug screening, is also provide a means of a phenotypic screen for senolytics. Again, but in this case, an unbiased screen for senolytics. And so again, using this cell line that I described before, where we have this tamoxifen inducible TRF2 deletion, what we did in this case is to color code these cells. And so in this case, we've made the senescent cells red through M-cherry expression, and the non-senescent cells green through GFP expression. And in this case, what we've done is to mix these cells together so that there's green and red cells at a known proportion, and then plated these cells in a 384 well format, and then used a novel chemical library of about, we've screened roughly 60,000 compounds. And so the idea is what you're asking for now is compounds that kill the red cells, but don't affect the green cells, right? So the red cells are senescent, the green cells are normal. These cells are isogenic for each other. The only difference is the deletion of TRF2. So they're the identical cells. One is senescent, one is non-senescent. We're asking for small molecules that kill the red, but keep the green around. And so and this is an example of, the first 13,000 of these compounds. And we're showing you high power images of the wells here of this 384 well. And so in the control well, you have a sort of standard mix of red and green cells. Again, the red cells are senescent, the green cells are not or normal. And the next lane over is navitoclax, which is a known senolytic, and you can see what it does. Again, the green cells are pretty okay, and the red cells are missing, and that's consistent with its known role in senolytics. And you can see that we're able to identify new molecules here, and they're shown in the box in the red, a bunch of molecules that have that property that they have, like navitoclax. They're killing the red cells, but they're not killing the green cells. And so they represent potential senolytic molecules. The problem is, in this type of screen, you don't know what the target is, you just know the activity. And so obviously it takes a lot of work then to go backwards and figure out what is it doing in the cell that provides this, ability to kill senescent cells and not non-senescent cells. But at least it's a start in terms of improving the armamentarium of senolytic drugs that might be available. So I'm talking too slowly, so let me just quickly go through some of the other things that we're trying to do or have done in terms of other approaches that we're taking. So one of the other things that my lab has been very interested in is developing mouse reporters that allow us to assess aging parameters in vivo. And so a few years ago, we developed this reporter mouse called Mitokema, which allowed us to look at mitochondrial autophagy or mitophagy in vivo. And it was a fluorescent reporter mouse that allowed us to do that. And shown to the right is in that orange is the level of mitophagy in the hippocampus of young animals. And to the right of that is the level of mitophagy in the hippocampus of old animals. And so this has been a very useful, I think, model for us and others to look at the process of mitophagy in living organisms. And I just want to say that we recently just published another mouse model that we're also very excited about as a platform for discovery. And we were very interested in understanding this concept. Well, I guess we were very interested in sort of understanding the serum proteome and really developing two things. One was spurred by this idea in aging biology that you can hook the circulation of old and young mice together in this thing called parabiosis. And there appears to be something potentially in the young serum that sort of rejuvenates the biology of the older mouse. But obviously serum is complicated. And so finding out what those molecules are, it's been very difficult. And so we wanted to get a platform that allowed us to do this in a more robust fashion. And this mouse has just been published, and we're sort of excited about it. And we're depositing it in Jack's lab for other people to work on. But we call it the Secreto Mouse. And it was developed, again, by two very talented people in the lab, Jie Liu and Jiang Yang. Because of time, I won't go through all the details, but it basically uses the concept of proximity biotinylation, which is this idea that you can there are certain enzymes that in this case derive from E. coli that in a sequence-independent fashion can add biotin molecules to proteins that are near it. And so we leverage that fact to express this proximity biotinylation agent molecule in the lumen of the endoplasmic reticulum. And what that did is that this molecule now tags with biotin proteins in the endoplasmic reticulum. And those proteins are the ones that, at least by conventional are secreted into the bloodstream. And so what Jie and Jiang did was to create a mouse with this construct in it that can be, which is flocked, and that can be turned on in a tissue of interest. And what that does when the construct is turned on and the mice are given biotin, proteins that are secreted then are tagged with biotin. And so the serum of these animals can be purified with streptavidin, and one can then identify what that tissue is secreting under normal conditions or under stress conditions. And so here's an example from the manuscript where we express this construct in the skeletal muscle of mice, and then exercise or did not exercise these animals, and then purified their serum, identified the biotinylated proteins in the serum by mass spectrometry, and then asked how those proteins change with exercise. And so this gives us a very nice list of proteins that are secreted by muscle under normal conditions and how those proteins might change with exercise. And we got some proteins that only we observed in exercise and other, so we able to sort of probe the tissue-specific secretome of mice. And I think for us, this was really, the real utility will be try to understand, for instance, how secretion changes in animals with age, but also in the really, again, in the rational development of biomarkers. So again, the ability to tag a specific cell or tissue and then in a mouse, and then cross it with a disease model allows you to see how, for instance, that secretion changes over the course of the disease. And then I think will allow for sort of, again, for just honing in on very complicated serum, just the cell or tissue of interest, and then seeing how that secretion changes, again, with that disease progression. Let me just wrap up very quickly just to say that the Aging Institute at Pittsburgh is also very interested in doing a variety of clinical efforts. And some of you know my colleague, Ann Newman, and it sounds like some of you were involved in the Energize trial as well. And there's a lot of interest in inflammatory markers and inflammation as a mediator of age-related diseases. And we've been very interested in that as well. And Ann and her colleagues, including Dan Foreman at Pitt, are anticipating the launch of a trial that we're calling the RIGHT study, which stands for Reducing Inflammation for Greater Health trial. And this is being done in as soon as sort of COVID abates in this area. And so we're going to do, Anne is going to oversee a randomized trial looking at whether or not a IL-6 antibody, clazakizumab, that's being provided by CSL Behring, is effective in lowering IL-6 levels in elderly patients, and whether in patients with higher IL-6 levels, whether it's effective in modifying, for the lack of a better term, sort of frailty sort of endpoints over a 6-month period. And so we're very interested in understanding whether or not reducing inflammation in this is causal to sort of a variety of age-related pathologies. And so that trial hopefully will start in the fall and has a number of endpoints that we can go over at some point. So let me just end by saying, I hope I've given you a sense of the direction that the Institute at Pitt is taking, and a little bit about what my lab and some of my colleagues are trying to take on. And we're really at the heart trying to go after the sort of the basic biology of age and try to understand what connects chronological aging or biological aging to a susceptibility for age-related diseases, and try to use that information to intervene with small molecules, or in the case of the right trial, with biologics, to sort of modify aging trajectory. I think all the data suggesting that aging is regulated, and that, like any other biological process, therefore it can be targeted by these approaching. And so I think, this is the approach that we're taking, but obviously from the talks that you've heard and from the literature and from the work that you're doing, there are many other approaches. And so I just wanted to share, again, with our strategy and the success that we've had today. So this is just a thank you to the people in the lab. As I said, Shahini is the one doing the CRISPR-based screen. Jia and Jiang had did the Secreto Mouse, and other people in the lab were doing other things that I didn't have at this time chance to talk about. So thank you very much for your time and attention, and I'll be happy to address any questions that you might have
Well, this was absolutely wonderful. Am I on?
Yeah, I can hear you.
Yes,
Okay.
go ahead, Alan.
It's really, this is a beautifully imaginative talk, and I understand why you wanted to go on after a brilliant career to have another brilliant career. A beautifully imaginative approach is to harness SASP and the high throughput selective cell survival screen, and then the tissue-specific Secreto Mouse. They're really tour de forces. I congratulate you, and I think we're all really lucky to have a chance to have you address us. So I'm going to take the bully pulpit here for a minute and ask a few questions, and then invite questions from other people. So people should, I guess, send them in the chat, right? Oh, it's in the Q&A. Okay. Well, I'll ask my 3 questions and I'll go to the Q&A. First question is, are the drugs that enhance TFEB and lysosomes acting, the drugs that you're checking anyway, are they acting through a common mechanism?
So yeah, so all of the drugs that are the small molecules that activate T-Fab all work through a common mechanism. And yeah, and it's an indirect mechanism, so they don't really bind to T-Fab, but we've been able to identify an E3 ubiquitin ligase that regulates T-Fab degradation, and that's the basis for how the small molecules work.
I see. So you're inhibiting T-Fab degradation?
Yeah.
Okay. And next question is, does T-Fab interact with Mtor at the lysosome? Are you affecting that interaction?
Not physically, but well, I mean, so yes. So the basic mechanism of T-Fab regulation, as I said, is its transport, its translocation from the cytosol to the nucleus.
Right.
And so that's under negative regulation so phosphorylation by Mtor is what regulates that process. So yeah, so there's a direct molecular connection between those two. So inhibition of Mtor results in the dephosphorylation of T-Fab and its ability to translocate to the nucleus.
Great, thank you. And my final question, at least I'll make it to final so other people have a chance to ask a question as well, is going back to your introduction, you talked about the x-axis and the y-axis. So does diminishing risk factors, say for one of the diseases, also affect the influence of aging on the disease, right? Because you can go both.
Yeah, I mean, I think of them, I mean, I think based on the epidemiologic, there's separate risk factors. I mean, I think all of those risk factors are independent risk factors. So cholesterol is independent from smoking, is independent from hypertension, is independent of age. So I think they're all, I mean, at least, I don't know the statistical test for proving that, but I think, you know, I think biological aging represents a separate risk factor than hypertension per se. You know, whether that's really true, I don't know. I mean, hypertension does it intervene on the sort of aging of the vasculature? I don't know what that, you know, I don't know molecularly what aging of the vasculature means. It's something that we're trying to understand, and I still don't really know if I have a concise answer for what, you know, tissue aging is versus overall lifespan aging.
Okay, thank you. So now we'll move on. Joel Mason asking, first he comments, as we all do, thanks for a great talk. And the constitutive upregulation of a cellular function as pivotal as autophagy could feasibly be detrimental. Have you observed unwanted side effects? Is it worthwhile considering target tissue specific upregulation or upregulation only to
so that's a great question. I think, as my mother used to say, everything in moderation. So I think that's certainly a reasonable concern. I would just say there's two things here. And again, and this has to go the particular mechanism of action of how our drug works. So we don't, I think if you constitutively turn on a pathway and you sort of ignore the sort of normal physiological activation pathways, then that certainly is really a big concern. Again, our approach is that we're not actually activating T-Fab, we're blocking its degradation. And so we require that the cellular tissue sort of turn on T-Fab, and we just keep it on longer. So we think that provides some degree of safety, and that we're not turning it on at all places at all times, but just keeping it on a little longer in places where the cellular tissue has said it needs to turn T-Fab on. So that's one sort of mechanism of regulation. There are malignancies that have been linked to overexpression of this family of transcription factors that T-Fab is, and that often leads to about 60 or 80-fold increase in T-Fab levels and activity, and that has led to cancers in the renal system and other places. Again, with this approach, we're getting sort of 2x activation of the pathway. So I think it's certainly a reasonable concern and worry, but we're hoping is that by the magnitude that we're turning it on and the way that we're turning on, that it won't be as big a concern for our approach. But obviously the data will come out over time.
Thank you. Larry Fike.
Hi, Larry.
He's here. Won't anti-aging drugs need to be incredibly specific if you're going to give them to healthy people for a very long time?
Yeah, specific and safe. So I think I think none of us are young enough or old enough to think that anything in our lifetime will be approved for longevity, nor I don't think, that's, age is not a disease for the FDA. I think our hope is that these central mechanisms are going to connect aging to age-related diseases, and so that these drugs will be useful for age-related diseases and not necessarily to make people live longer. So I think any sort of drug that we or others develop will really probably have to be for a specific condition and not necessarily for aging. With that said, for the IL-6 type approach, we do think that, and we're attempting to meet with the FDA about this. We do frailty per se is a very tough thing talk about. But in the patient population that the right trial is going against, is targeting, these are elderly people over the age of 70 with high IL-6 levels, and that patient population epidemiologically, is and they have impaired walking speed. That's sort of a sort of entry criteria. So we kind of feel like that if you can improve, what we were interested in talking with the FDA is that if you can improve walking speed, for instance, in this segmented population of the elderly, whether or not that would be, ability, something like that would be enough to meet registration of an agent or not, or whether or not you really have to show something else. But even in this patient population, if you don't do walking speed, they have a very high rate of morbidity and mortality. So this population, based upon work in the energized other, has a high risk of going into the hospital. So there are sort of hard endpoints maybe that one could think about for intervention that are not overall lifespan, but are involve quality of life or hospitalizations that one could think about in a sort of predefined age cohort.
Thank you. Chris Wiley notes exciting work, Toren. I guess you must know him.
Yeah, hi.
You're
Is he
Chris.
at Tufts?
He's at Tufts now.
Okay.
Right, newly arrived, and we're delighted to have him. Your CRISPR design suggests you might also detect modulators of senescence, such as immortalization or escape from senescence due to overrepresentation of guide RNAs. Did you detect anything interesting in this area?
Yeah, we saw nothing escape from senescence. Nothing. There's nothing that grew out of this the population. So I don't think it doesn't look like a single gene deletion can rescue this form of can overcome this form of senescence. So so no, we did not see anything that escaped from senescence.
And another question, Stefano Pizzarini Arani is asking, is aging the overall complex final entropic outcome of biophysical friction?
Well, that's what I grew up thinking.
Right.
But that would, I mean, it's sort of semantics and tautology to some degree, but
right.
I can't say no, but then I would argue, why can you alter its trajectory by changing a single gene if it's completely stochastic? So I don't know. I mean, my answer is, I think it's regulated, but I don't know exactly what we're regulating. So when we change Mtor, I don't know what Mtor is doing that changes lifespan and changes health span. So I can't say that we're not changing something that's stochastic in nature. But to me, it doesn't seem satisfying. So I'm going to say it's I think it's more regulated than stochastic than I used to believe.
Thank you. So let me ask another question, going back to Joel's question and maybe some of the original points. Are you learning from your manipulations, say of things like T-Fab and upregulation of autophagy and the limits of autophagy? Are you learning about the biological limits of how far you can go to turn on and turn off specific pathways with salutary effects? Or when do you incur damage? And is that like tissue specific?
Yeah, obviously, as part of getting a drug to the market, you have to do a lot of tox studies, and so we're in the process of those studies, and we do see, as we raise the concept of the drug, we don't see any overt toxicity. The animals seem fine, but when you look histologically, you do see things that look like lots of autophagosomes in the cell, and that's a toxicological finding. And so there might be a point in which if you whip this pathway too much, the cell, I mean, obviously these things have a homeostatic balance, and in normal animals and normal people, you don't, your autophagy is set to take care of what it needs. I think as we get older, for reasons that are not entirely clear, this pathway degrades. And so I think the effects in elderly people might be beneficial if we could restore it back to what it was in youth and when you were young. I don't think there's going to be that much benefit for turning these things on in young people, except for some rare examples where the situations in which, you know, they have specific diseases where quality control is really important.
Okay, thank you. Well, another question just came in, Shruti Sharma. Great talk. Perhaps you addressed this and I missed it. Are you planning any parabiosis experiments with the Secreto mouse? And
No, we aren't. I mean, I find those experiments really seem cruel and medieval to me. I don't know. We're not doing that, but we are doing a lot with the animal, and we're really very interested in this biomarker. I mean, to me, biomarkers are sort of a underdeveloped field in terms of sort of rational platforms, and so, you know, we're very interested in trying to use this as a way to rationally go after biomarkers for disease. And so I'll give you one example that we're excited about. Well, we have no data, you know, is pancreatic cancer, where, you know, obviously most of the time people get diagnosed and it's really too late. And there's some beautiful genetic models of pancreatic cancers in the mouse where you can turn on knock out P53 and turn on KRAS in specific cell types in the pancreas. And those go through this very sort of stereotypical development of a sort of precancerous lesion and then a cancer lesion. And so we think we can express our reporter in the same cell that we're turning on P53 and KRAS and use that to sequentially monitor what's being secreted by that specific cell, both at the very beginning and in the precancer stage and then in the cancer stage, and use that basically to track, you know, to sort of take away the 5,000 other proteins that are in the serum that are not coming from the pancreas, and really hone in on the cells that are genetically modified by P53 and KRAS as a way of looking really early on in that disease, what we can detect in the serum. So I think those are the approaches that, you know, which are not really aging related, but I think are really the sweet spot for that type of mouse.
Mm-hmm. So as a follow-up, are there good studies suggesting that a specific type of cell influences the youthful secretome?
No, I think the studies on secretome in humans has been, you know, sort of, you know, taking serum and doing really nice mass spec identification of what changes.
Mm-hmm.
And so people have developed, you know, sort of nice, you know, PCA plots of, you know, young versus old serum and what changes and what's driving those differences. But where that comes from, I think is very difficult. You know, the analogy I use is like, you know, it's a highway and there's all these cars, you know, the serum is a highway with multiple cars. And, you know, what you need is a license plate to figure out where the car came from. And you know, and if you don't have that license plate, all you say is there's lots of cars here. And, you know, it's either busy or, you know, we have different types of cars, but you can't say where that car was coming from. And this approach is one way of giving each protein in the serum or proteins in the serum a license plate because they only have biotin if they're coming from the tissue that you've tagged.
Okay. Well, I think we only have 1 more minute left of your time before you've got to run off. So I want to thank you really for a brilliant talk. Thanks so much.
Thank you so much, Alan. Thank you, Roger. And thanks everybody for listening.
Thank you, Toren. It was an excellent talk and really great to hear about all your exciting work. So we appreciate it. Thanks, everyone, and thanks for attending today. We had almost 80 people on our seminar today, so it's great.
Wonderful. Thanks.
Okay,
Enjoy the weekend.
thanks.
Thank you very much.